Okay, good morning, everybody. It's my pleasure today to welcome everybody to the next installment of the Early Career Immunology Seminar Series, where we try to highlight some really outstanding young investigators who are
Philip, you're muted.
Sorry, I was just getting a feedback. Sorry. My name is Philip Ahern, and I'm an assistant professor at the Lerner Research Institute, Cleveland Clinic. And today it's really happy to introduce Billur Akkaya, who's an assistant professor at The Ohio State University. So Billur began her academic career with an MD at Hacettepe University in Turkey. And following this, she moved to the University of Oxford, where she performed her DPhil studies under the supervision of Simon Davis and Richard Cornell. And there she was funded by a Hugh Pilkington and a Felix Scholarship. Following this, she moved to the United States and performed her postdoctoral work with Ethan Shivak at the NIH. And I think it's here that her fascination with Tregs and how they operate was initiated. And a few years ago now, she moved to the Ohio State University to set up her own independent laboratory, where she's continuing in her efforts to try to define the antigen specificity of Tregs and the molecular mechanisms through which Tregs are able to mediate their immunosuppressive functions. So I don't want to take up any more of her time, but just to remind you, if you have any questions, please post them in the chat box and we'll get to them at the end. So with that, Billur, welcome, and I'm looking forward to hearing what you're going to talk to us about.
Thanks very much, Philip. It's a great pleasure to be here addressing to you guys. Well, I wish these platforms are more and more physical in the future. So it's a great honor to be invited to become one of those early career investigators who are presenting here. So as Philip has already told, I am an assistant professor at the Ohio State, and I am looking into molecular mechanisms of regulatory T cells, and I have a passion in looking into the Treg synapses, which kind of forms the content of my talk. So I will try to tell you about what I do, and I try to give a glimpse of our unpublished data. So most of the things I will present here, at least 2/3, will be unpublished. So I am hoping to get your wonderful comments and questions about it later. So in the first part, I will talk about how Tregs work. I mean, what we know so far about how Tregs work. And then I would like to get into how I try to address this question by looking into Treg phosphoproteome and proteome. And also we have some evidence suggesting that TCR co-receptors play an important role in the Treg antigen-presenting cell synapses. And in the second part, I would like to talk to you about some of the crazy aspects of being an immunologist at the crossroads of, mouse immune system and human immune system. And it's not an easy task to address questions about antigen specificity, in particular with the Tregs, but we will get into it. Okay, so this is something I like to state, which is, quite dramatic. Tregs only form 5 to 10% of peripheral CD4 positive T cells, and they are identified by their expression of master transcription regulator FOXP3. And this 5 to 10% really controls the 90 to 95% of CD4s and also CD8s. Yet, their ablation is quite dramatic. So this is a graph showing the situation in a FOXP3 diphtheria toxin receptor knock-in mouse model. So compared to wild-type mouse, when you start treating these DTR mice with diphtheria toxin, you see that all the mice, so these are adult mice, so they have Tregs throughout their lives, and all of a sudden when you start treating them with diphtheria toxin and ablate their Tregs, they start dying, and 100% of them dies within 20 days. And this is mostly due to autoimmunity. So this means it is important to understand how regulatory T cells work to prevent autoimmunity. But on the flip side, we know and we appreciate more and more that Tregs accumulate in the tumor microenvironment and hinder anti-tumor immunity. So we know that they first see the tumor antigens in the tumor draining lymph nodes, and they upregulate chemokine receptors, especially CCR4 and CCR8, move into tumor tissue, tumor microenvironment itself, and they undergo several metabolic adaptations. And this is, while this is a hostile environment for effector T cells, Tregs really thrive there. So there are many mechanisms proposed for why they thrive there. For example, effector T cells may become induced or peripheral Tregs, or there's an increased survival ability for the Tregs over effector T cells. Bottom line is, this also makes us quite wary of, tapping into Tregs, their mechanism. So if we try to, boost them in autoimmune diseases, it may come with the expense of promoting pro-tumor activity of regulatory T cells. Molecularly, independent of any disease, it is important to understand how regulatory T cells work in the first place. We have shown, so this is my postdoctoral studies were mostly about understanding at the, antigen-specific level how regulatory T cells work. And one of the things we found it was quite dramatic that antigen-specific TRex can capture peptide MHC2 complexes, they are cognate complexes, and they inhibit effector T cells that have the same peptide specificity, while the T cells that don't recognize the same antigen could still get stimulus. And there are many other mechanisms that have already been proposed. We call them bystander mechanisms. And we know that Tregs can also downregulate or capture CD80-86 molecules. And they also can be an IL-2 sink and consume IL-2, which kind of reduces T cell survival, and they secrete IL-10, TGF-beta, IL-35, all these immunosuppressant cytokines, as well as they can also be anti-inflammatory in the way that they cleave extracellular ATP and then they turn it into anti-inflammatory adenosine. And many more. So we still, we are in the process of adding to this list. But I would like to show you some of the key experiments where we obtained that they work in an antigen-specific fashion, and it is quite dramatic when you match the antigen specificities of regulatory T cells to that of effector T cells. So there's a disclaimer. So in these experiments, we tried to address the antigen specificity and we created an environment. So this is we use a very reductionist approach, and we sort of limited all the effects that the inflammation or cytokines can add to this puzzle. So what we did was essentially we used TCR transgenic mice as a source of naive T cells, as well as a source of regulatory T cells. So we had two different antigen specificity, Tregs and naive T cells. And the experiment was we mixed these different specificity naive T cells and gave them to a mouse that was also injected with dendritic cells that were freshly isolated from a normal mouse and loaded with 2 different antigens. So the idea was giving these dendritic cells through the food pad, we generated an immune response at the popliteal lymph node. This is at the back of the knee. And the T cells that were injected IV, they would recirculate the body, see their antigens, stop at the popliteal lymph node, and unleash an immune response. So this is in the absence of any, antigen-specific Tregs around. And we asked the question, so in this mouse, which has, double pulse dendritic cells, 2 different antigen specificity T cells, what happens if we introduce one Treg, one specificity Treg, or the other specificity Treg? It was quite dramatic what we see. So in the control case, these rows are showing you the naive OT2 T cell proliferation versus naive SMARTA S T cell proliferation. So in the control situation, there is no Tregs around and the cells happily proliferate. So we check this by CFSC dye dilution. And in the condition where we added all Tregs into the system, we saw that ST cells were still happily proliferating while OT cells were inhibited, even though these DCs were, contacted by this OT-reg. DC wasn't globally inhibited. Same was the case. So we tried the opposite way. We gave ST-regs and we looked into the proliferation, and we saw this time ST cells got inhibited, but OT cells were proliferating just fine. This was black and white antigen specific and quite puzzling, and we tried to understand why this is happening. And in a set of experiments where we looked into the peptide MHC2 complexes on the surface of the dendritic cells and the Tregs, we saw that Tregs perform this by peptide MHC2 theft. And this theft is quite specific, that even though the Treg binds dendritic cell, it doesn't capture the other antigen that is already being presented on the DC at the same time. And we saw that. So this is, again, a 2-antigen, 2-TCR transgenic Treg system. The reason why I am now using H versus M Tregs is that we have reagents to detect the peptide MHC2 complexes. So this Treg is specific for HEL, IAK complexes, while this Treg is specific for MCC, IEK complexes, and there are antibodies that were described years and years ago, more than 20 years ago, that we used to detect the peptide MHC2 complexes. So cutting the long story short, we saw that while these Tregs capture these complexes specifically, there was also a specific reduction on the level of these complexes on the dendritic cells. So this is a co-culture experiment that I am showing the data from, but we did this with in vivo adoptive transfers, and we did this with iTregs versus TTregs, and we saw that this happens in this system, which is kind of reducing the dendritic cell presentation of antigen in an antigen-specific manner. And just to stress that these dendritic cells are double pulsed, so they present both antigens at the same time. And while HTreg doesn't reduce the M complexes, MTreg reduces the M complexes. So hopefully I was able to convince you about this stealing phenomena. And we were quite intrigued, and this kind of forms the basis of my current research program. I dedicated my research program here for the first 5 years to understanding why do Tregs steal peptide MHC2. And what makes Treg synapse unique, that when we compare Tregs to effector T cells don't do this at the level that Tregs do. So Tregs they capture complexes, and dendritic cells are wiped clean, while effector T cells can also capture complexes at a lower level, and they don't wipe dendritic cells clean. So as I said in the beginning of my talk, it's not easy task to really ask these questions. You really need to use verified systems and create your own tools to be able to ask this. So mostly the easiest way to obtain these antigen-specific TRegs is using TCR transgenic FOXP3 reporter mouse systems, where you can isolate naive T cells and differentiate them in the presence of anti-CD3, CD28, and also TGF-beta into Tregs. And you can go ahead, sort these cells out for GFP, but we have methods to generate these with 90% purity. But you can get a pure Treg population, and at the same time you can create your control cells, such as activated T cells. So basically these cells are activated in the absence of TGF-beta, and we just ensure there is no endogenously produced TGF-beta that is active on the T cells by adding anti-TGF-beta to this culture. Alternatively, you can inject these mice with IL-2, anti-IL-2 complexes to boost the number of Tregs, because these mice don't have a normal repertoire. And if you go ahead, try to isolate fresh Tregs out of these mice, the numbers are pretty low. So you need to boost them with something. So in my hands, this regimen quadruples the Treg numbers. So you can get those Tregs that way too. And in human systems, okay, it is trickier in human. So there is one way we are now trying to use to get our hands into human systems too, which is sorting conventional T cells and Tregs out of the bloodstream, out of the PBMCs, and adding them, doing lentiviral transduction to add them our TCRs of interest. So this is another way that we are using. So I will start by showing you some interesting things we have come across in our previous studies with mouse Tregs. So one of the things I wanted to do was to understand how Tregs interact with the dendritic cells, because they are performing quite unique activity, which is different from the effector T cells, and which kind of leads to peptide MHC2 theft. However, upstream of this, there is the interaction that is happening in the first place. So one of the ways we tried to understand this was just simply looking them under the microscope. And we did this microscopy at multiple levels. We did scanning and transmission electron microscopy, confocal microscopy. We use our staple flow cytometry. We did intravital microscopy. So we did microscopy at multiple levels. And I will go one by one about what we found. So it was quite striking to see them in action under the microscope. So this is coming from our SEM sessions. So this is performed within 2-3 hours after Tregs were co-cultured with dendritic cells, and our control cell types were naive T cells, and these all were derived from the same TCR transgenic mice, by the way. So these have the same TCR affinity, and this is an TCR avidity control, and this is our iTreg. Looking at them, the morphology of naive T cells was more or less round when they bind dendritic cells. So we saw, you can come across this picture in the literature anywhere else. So the round morphology T cell is interacting with dendritic cell. No surprises there. And activated T cell, it was bigger. It was pricklier, so we saw more microvilli on their surface, but they preserve their round morphology more or less when they are coming in contact with dendritic cells. However, we saw something quite dramatic in Tregs. They looked like amoeba. So this is the red ones are Tregs, while they are contacting with dendritic cells. So they had prominent phylopodial extensions to dendritic cell synapse. And also we saw something suggestive of uropods. So this is a structure that is similar to a suction cap. So basically, this is responsible for So this is a result of very dynamic actin reorganization inside these cells. And when we had a close-up view to the binding interface between all these cells, we saw that. So this is as much as we could resolve to this area. So we had to do a tilted SEM to be able to resolve into this. And this is, highlighted zones are where we have the little microclusters, which supposedly have the TCR and the co-receptors. And in the naive T cell synapse, we see that this is rather, multiple of them. And if you take the width measurement, they are not too wide. So there is multiple of them. And in the activated T cell synapse, these microclusters kind of conglomerate. So they come together, which is suggestive of a more mature synapse structure. And when we looked at the Treg interface, it was again quite dramatic. We saw less of these domains and they were wider. So it was suggestive of a more organized, even more organized than activated T cell, more organized synapse structure in the Treg dendritic cell synapses. And these are the morphological adaptations that also corresponded to peptide MHC2 uptake. So within the same time frame, again, if you try to track MHC2 complexes. So this is a confocal microscopy image where we looked at total MHC2 that originated from dendritic cell and ended up on regulatory T cells. This is 3 hours, about the same time we did the SEM imaging. So you can see that MHC2 positive patches, membrane patches, are originating from dendritic cells and ending up on Tregs. And if you would like to look at the antigen-specific peptide MHC2 complexes and the movement of them, we weren't able to do that with confocal. I think the sensitivity wasn't quite well. So when we looked at it by transmission electron microscopy, and here the quantum dots, so the arrows point to quantum dots that label the specific peptide MHC2 complexes. This is dendritic cell. It is, you can't really say that these are moving inward or outward, but we are thinking that these are moving to the synapse. So these complexes are being deposited to the synapse, and these are actively being taken up by Tregs into structures that are similar to early endosomes. And you can also see isolated patches of quantum dot positive membrane areas on the Treg surface. And this is just a quantification effort. It is not uniform, obviously, but you can detect that the quantum dots are only acquired by Tregs within this time frame. And from up to now, I try to present our published data, mostly to give you an overview about the interesting things we come across in terms of Treg dendritic cell synapses. Now I will try to show you some of our unpublished data and efforts to identify what is going on in the synapse. And one of the questions we wanted to ask was, sorry, one of the questions we tried to ask was, what is the role of cytoskeleton in this? So it was, you know, in a sense, it is quite obvious. It is important. Cytoskeletal movements and reorganization is important in any T cell. So we can't really distinguish, you know, whether, you know, cytoskeletal reorganization is important in Tregs versus effector T cells. The answer is no, it is important across the board. But we had some effort in understanding these elements that we know play a role in the T cell synapse. So there is quite a bit of literature in terms of all these, you know, adapter molecules, what is linking TCR signals to the cytoskeletal elements. So we wanted to do a quick experiment. Well, it wasn't quick because we were trying many inhibitors. So what we did was we isolated our Tregs and we pre-treated them with specific inhibitors. And we washed them rigorously, and then we co-cultured these Tregs with dendritic cells. And after 18 hours, you can get a nice readout with flow cytometry. So we looked at the peptide MHC2 uptake by Tregs. So the reason why we didn't just dump these inhibitors into the co-cultures was because these inhibitors are not selective, so they can as well inhibit dendritic cell cytoskeletal rearrangement. So to our best effort, we were able to look into small GTPases that link TCR signals to cytoskeletons such as RHO-A, RAC1, CDC42, and we were able to get a dose-dependent reduction in the amount of peptide MHC2 that is captured by regulatory T cells. Secondly, well, this is rather an arbitrary grouping, but we looked into many things, such as you know, the elements that are known to play a role in actin movement, arching, and contraction of uropod, maybe centripetal movement of the TCR and the coreceptors, and filamentous actin, and also stabilization of synapse and integrin functions such as tailin, that is kind of responsible for the LFA1 function, conformational change in function, esrin, facin, all these molecules. And, you know, across the board, it suggested that the cytoskeletal reorganization is an important part of this puzzle. But we need to take these findings with a grain of salt, because these agents, the small molecule inhibitors, can simply paralyze these cells. So you wouldn't think that, you know, okay, this T cell is Treg is going to do whatever it is destined to do, but it is just inhibited in this little you know, cytoskeletal part. That's not the case. These may paralyze the cells. So we have to come up with some more you know, less intrusive ways of addressing what is going on in the Treg synapse. And one of the things I wanted to do all along was looking at the phosphoprotein of these Tregs that are interacting with dendritic cells. And the reason I have always wanted to do it, but I never got to do it, I didn't have the expertise and the skill set. And one of the nice things, starting your own lab and ending up in an institution that has a lot of cool people, I was able to find a collaborator, Brian Searle. He is in the Immuno-Oncology Institute. His lab is downstairs to my lab, and I just broached the subject. Hey, you're a phosphoproteome expert, so I have this question. Luckily, he was equally interested, and we decided on taking this effort jointly. So the system is basic. It's not no different than I told you earlier. So basically, we are obtaining our antigen-specific Tregs and obtaining our dendritic cells, co-culturing them. And okay, then things start getting complicated because there were, I'm an immunologist, so I was thinking that, okay, so at this point, if we can fix the cells and fix the phosphoprotein, phosphorylation events at the same time, and we can go ahead and sort the cells apart and then run them one by one, like DCs and Tregs separately in the mass spectrometer. So I came up with this Metamorphixation idea, which I use quite often for phosphoflow analysis, which works fine, for fixing and permeabilizing cells if you're doing some, looking into some phosphosignatures by flow. The short answer to my question was quite dramatic. So it didn't quite work quite well. So the co-culture system, so basically, this is a co-culture system. So when you feed the preps into mass spectrometer, you wouldn't get a distinct, answer about where the peptides are coming from. So it can be DC, it can be Treg, so it's just a mix. So being able to sort them would have been nice while preserving the phosphoproteome, but we saw that even the proteome, so this is just a quick analysis for the methanol fixed cells versus snap frozen cells versus rested cells. So rest period is supposed to mimic the sorting period of cells. So basically, bottom line is methanol fixed ones had a different proteome. So we said, okay, this is not a viable option, so we have to change gears here a little bit. The next thing we tried, we are still working on this. So there is also another thing you can do. So you can basically grow your dendritic cells, differentiate your dendritic cells from bone marrow precursors, and you can feed them with different molecular weight amino acids. So this is called stable isotope labeling with amino acids in cell culture, Cylac in short. So the Cylac labels you can use are, heavy arginine, heavy lysine, medium arginine, lysine, and light ones. So basically, this allows you to barcode the cells and make your prep as a co-culture. Start from the co-culture, make your prep, and feed this into your mass spec. And once you get the peptides, they will be either heavy or light, or heavy or just medium. You would be able to tell them apart. Where what are the sources of cellular sources of these peptides. So we are still in the early phases of being able to do this. People who use BMDCs also know that BMDCs, how much they resemble conventional DCs, what are they, what do they express? These are all still ongoing, questions in the field. So we are trying to do our best to generate them in the presence of GM-CSF IL-4 or FLT3 ligand, while also depleting B220 positive cells to eliminate plasmacytoid dendritic cell generation. So, but this is about 10-day procedures, which allows us to incorporate these DCs, our heavy and light amino acids. So this is just a crude analysis showing that our 10 grown DCs versus Cylac, light Cylac grown DCs are in a sense similar, and T cells are just the control here. It is their proteome is very different. But this is not the analysis that we would like to do. We are also looking into their growth and differentiation. So we were able to get a similar differentiation with our normal protocol with R10, light and heavy. So it's about 65 to 70%. And you can sort on this population before you proceed with co-cultures. And this is again a very early analysis from the Cylac Incorporation of these BMDCs. So in order to incorporate Cylac, you have to do protein turnover. So you need to be making new proteins so you can incorporate all these amino acids from the media. So this is more Brian's domain, and I will try to tell you how we look at Cylac Incorporation. So basically, we grow the BMDCs either in light amino acid media versus heavy amino acid media, and we take so this is I was told this is how it was done. So you need to take a baseline with the light media, and you check and then you mix your preps 1-to-1, light and heavy preps 1-to-1, and then this is where you check your heavy protein incorporation. So this is a graph showing that 42% of the proteins do not turn over, and just keep in mind 50% of this prep is already coming from light amino acid fed BMDCs. So, and half of the proteins incorporate at least at 38%. So this is the level of integration, 38%, which is telling us that, and Brian is confident that any integration level above 30% would be okay to proceed with. So this is really hopeful. So we are really looking forward to proceeding with the co-cultures because our incorporation is suggesting that we are doing this at a good level. And this is mostly coming from light amino acids. So half of proteins is kind of an underestimation. So this is not really half of the proteins, basically. So if you think about the light amino acid incorporated ones, it is much more than half of the proteins incorporate at least 38%. So this is so far our efforts for doing our Cylac to be able to barcode the cells for future phosphoproteome analysis, and hopefully it is going to give us some results that we are looking for. And in the meantime, we have some targeted efforts to understand the role of core receptors and also the role of some adhesion molecules and integrins that play a role in immune synapse. And earlier, we have shown that in addition to peptide MHC2 molecules, TRegs also acquire ICOS ligand, CD86, and PDL2. And we are trying to understand also whether these molecules really contribute in a positive or negative way to the ability of Tregs to capture peptide MHC2 and function. So these are the ligands that are captured, and we have already addressed the possible role of CTLA4 in Tregs' ability to capture peptide MHC2 complexes. So CTLA4, it's constitutively expressed by Tregs, and we know that it's a prominent player in Treg function in vivo and in vitro. However, when we plugged this in our antigen-specific system, it didn't work quite as expected. So if you delete, so I was able to get my hands on antigen-specific CTLA4 knockout TRegs, okay, and these TRegs were co-cultured with dendritic cells that present antigen, and I was able to compare CTLA4 wild type versus CTLA4 knockout Tregs, antigen-specific Tregs, in their ability to capture peptide MHC2 and also suppression. So the bottom line is these Tregs are as suppressive, to our surprise. These Tregs are, first of all, as suppressive as the wild type counterparts. I'm talking about the antigen-specific ones. And I show that they capture peptide MHC2 just fine. And they suppress just fine. So it was quite puzzling. Blocking or taking away CTLA4 didn't really change anything in this phenomena of peptide MHC2 theft as well as suppression. And after this surprise and shock, I tried to address some other key molecules. So, you can buy these antibodies, such as blocking anti-CD28, ICOS ligand, PD-L1, PD-L2. So this is the readout is, again, peptide MHC2 capture, and this is the level that Tregs are capturing without any treatment. And I didn't see anything significant happening when I blocked any of these B7 family molecules. And this is surface level of the peptide MHC2 acquired by regulatory T cells. This is looking into the intracellular portion of this peptide MHC2 in the regulatory T cells versus effector T cells. Nothing really. And we try to understand the role of co-stimulation overall. So, okay, the Tregs seem to be acquiring some of these molecules, but whether this capture of B7 family molecules have any importance in the global DC function. So, in other words, stripping off dendritic cell of these molecules would have any consequences on this DC's ability to activate, any T cell. So there is so we don't know the extent of this capture. We know the capture happens, but we address this in a way that we can really ask this question. So what we did was we co-cultured double pulsed dendritic cells with Tregs, and then sort these Tregs the next day, remove the Tregs from the environment. So now that the dendritic cells were pre-treated with Tregs, and whatever is taken has already been taken. And then we cultured these dendritic cells with fresh, naive T cells of either specificity, and we were able to ask the antigen specificity question here in this pre-treatment assay. So the dendritic cells that were just that spent overnight without any Tregs and co-cultured with fresh naive T cells the next day, they just gave enough stimulation to those fresh T cells that those cells kept on proliferating, so they were able to proliferate. When we pre-treated those dendritic cells with all Tregs, those all T cells were the ones that were inhibited, not the ST cells, so which is suggesting that there is removal of antigen-specific elements, but not a significant removal of the B7 family molecules, or DCs are able to make more of those upon removal. That didn't really affect the proliferation of the other cell type. DC function was globally okay, except for the antigen part. And vice versa, dendritic cells pre-treated with STregs were still able to stimulate OT cells, and ST cells were inhibited this time. So I know this may be quite puzzling. So they capture a little bit of co-stimulatory molecules, co-stimulatory ligands, but that level of capture is negligible. We still need to understand that. And looking at the DC levels of these molecules, there is still heaps of B7 family ligands, but it is a little bit reduced, which probably doesn't give rise to any phenotype in the ability of T cells to get stimulated. And also, if you look at the mRNA levels of these molecules, they are intact. So the mRNA is still produced. So probably DC is able to make more of those. Anyway, so I will switch gears in our targeted efforts furthermore to tell you about our preliminary findings on another molecule that we think that is important in the Treg synapse. So this came up from just a literature search or just plain knowledge from T cell biology field, that LAG3 is a CD4 homologue that binds MHC2 outside the peptide binding groove. And it was kind of, this was an obvious candidate that binds to MHC2. So I thought, okay, well, let's ask the question whether the LAG3 has any role in the Treg synapse in terms of capturing peptide MHC2. So I obtained the mouse, crossed it to the antigen-specific background, too many crosses, and also I like to keep my reporters in the system. And it was interesting, and this was along the lines that which has been previously shown in OT2 LAG3 knockout T cells. So this is not really new, but I basically demonstrated the same thing. The antigen-specific TRex that lack LAG3 were less suppressive. And looking at the peptide MHC2 capture, there was I was able to test the wild type versus heterozygous versus full homozygous knockout Tregs. And I saw that there is a dose-dependent reduction in the ability of Treg to capture peptide MHC2 when LAG3 is lacking. And looking at the DC surface is just another view to look at things, how the peptide MHC2 is stolen. So in the knockout Tregs, peptide MHC, the level of peptide MHC2 is higher compared to the level of peptide MHC2 on DCs when you co-culture DCs with wild-type Tregs. However, just as a note, it doesn't completely abolish the ability of Treg to capture peptide MHC2. So probably there are other molecules that play a role along with LAG3 in this setting. And we like to do microscopy with all these co-culture systems as well as intravital microscopy, but this is coming from a co-culture assay where we looked into the Tregs that are wild type. So here the Treg is blue, blue, it's the intracellular, it's just the protein staining. And the surface is stained with a CD4 antibody, which is green. So this is the CD4 membrane, CD4 positive membrane. And the sun-like structure here, it is the DC cytoplasm, and the membrane is labeled with a lipophilic dye. So basically, we are just simply watching what is going on. So Treg comes and binds dendritic cell, pinches the DC membrane. It rolls around, and lifts up with a piece of DC membrane on. Looking into the LAG3 knockout T cell, it binds the DC just fine, and binds and rolls, tries to pinch, but it doesn't really take off. So, and we looked at the lipophilic dye intensity that is picked up differently by these two types of Tregs, and it is nice looking at these, but the quantification also shows us that indeed Tregs aren't able to lift off with a membrane, with a pinched membrane that originates from DC. And this is just a flow cytometric image from an identical culture, looking at the PKH26 uptake by knockout versus wild type. So it is considerably less. We have some crazy questions, such as, okay, LAG3 is not a Treg exclusive molecule. It's also present in, so it is also upregulated upon TCR stimulation in effector T cells. And we know that it has some inhibitory role in, exhausted cells also express LAG3. So we just wanted to ask, okay, so Tregs, deleting LAG3 reduces function. What happens in effector T cells? So effector T cells can also, interact with dendritic cell, pinch off some part of membrane. And this is also a viable readout to look at in terms of the ability of activated T cells in this experiment to capture membrane parts from DC. And the interesting thing is, and I am leaving you with this question really, activated T cells, at the starting point when LAG3 is intact, they are acquiring the membrane parts similar to the knockout Treg. And when you delete LAG3 on activated T cell, it seems to be a reversed phenomena. It comes to the level of a wild-type Treg, so it gains the ability to capture more and more DC membrane. So there is a reversal of this phenotype, and we are still trying to figure out why this is happening. And there is a reversal of the phenotype that we are seeing between effector T cells and the regulatory T cells. And happy to hear your thoughts on this. And the other thing is, I told you earlier that blocking any of the B7 family molecules doesn't have any effect on their own. So I tried to throw in all these blocking antibodies in a co-culture where I co-cultured LAG3 knockout Tregs with dendritic cells. And quite dramatically, I saw that in LAG3 knockout Tregs, remember, they were still able to capture a tiny little bit and deplete so the effect wasn't in full. If you block CD28 signaling in those cells, you would abolish peptide MHC2 uptake completely. So now we are really putting our fingers on LAG3 binding to MHC2, maybe exerting a pull that is optimal for Tregs to capture, as well as the role of CD28 CD86 interactions in terms of giving Tregs the ability, the unique ability to they are giving Tregs aid in terms of helping LAG3 to capture peptide MHC2. So the future directions, the conclusions and future directions, I will keep it simple. We are still addressing the possible synergy between LAG3 and CD28 family of receptors. And we are trying to also understand what is giving rise to this, different phenomena in Tregs versus effector T cell synapses. And there is something called a post-translational modification that LAG3 is subjected to. It is called LAG3 shedding. So we are trying to understand whether LAG3 shedding plays in a role in this. And we still continue our efforts to understand the Cylac and the phosphoprotein aspects that would give us some information about the Treg DC synapses, and hopefully we will have some more targeted efforts to delete these B7 family members in primary cells using CRISPR-Cas9 system. And very briefly, I will touch base on our human systems for understanding Treg biology and specificity. I'd like to show this, and I will finish a couple of slides. I only have a couple of slides more, and I would be happy to take any questions about it. But I would like to share with you some really preliminary data but it is really cool that we were able to show in the human systems that Tregs are able to do a similar thing to the mouse Tregs that we have previously worked on. So here, I have a collaborator from University of Florida. So he's an immunologist, Todd, and he is a great type 1 diabetes researcher, and he has his hands in a lot of things. So he works on, so he has described a method for lentiviral transduction of Tregs that are obtained from PBMCs. And he has access to type 1 diabetes patient PBMCs with a risk allele. So basically, he had all the tools that I needed to apply our knowledge in mouse Treg biology to human Treg biology. But the system is again rigged. So these are again artificial, somewhat artificial, not super physiological Tregs or effector T cells. So they were introduced TCRs with known specificity that we know the peptide for, and we know the MHC2 risk allele. So basically, we create a co-culture system that is identical to what we did with the mouse. And very briefly, we showed that the Tregs in this system were able to capture peptide MHC2 from B cells and monocytes at a higher level than effector T cells. And we were able to ask the question about affinity. We show that it increases, the capture increases with affinity, but across the board, either low or high affinity, Tregs capture more than effector T cells. So this is what we try to understand. So basically, this shows us the system also kind of works in human, and we can use these TCR transgenic human Tregs and effector T cells as tools to ask other questions that we would like to ask, to resolve into the synapse, look at the phosphoprotein. So these are all possibilities that we will think about in the future. Another effort I just initiated is also understanding the bona fide self-reactive Tregs that circulate in our bloods. So it is nice and all using TCR transgenic systems, but you can ask the question, I mean, this is not real. You just make this, as a tool, which is right. So we are now joining efforts between OSU and University of Florida to start collecting also OSU samples and analyze them fully in terms of the single cell RNA-seq, their transcriptome, single cell transcriptome, and also paired alpha-beta sequencing to identify the clones, the Treg clones and the effector T cell clones that oligoclonally expand in these patients. And we would like to also use our own efforts to identify, maybe get these PBMCs, add them peptide, and try to look into some markers that the tissue reactive, those antigen reactive Tregs can express. There is some information from the allergy field that suggests a quick co-culture with antigen may help antigen reactive Tregs that are already present in the PBMC mix express CD137. And we will look into this possibility in the autoantigen setting to see whether we can use anything in flow cytometry approaches to identify the antigen reactive bona fide Tregs. And the other thing is, very briefly again, we will try to put this TCR alpha beta information in use. So we are also devising a new platform. This is an antigen discovery platform where we create antigen-presenting cell library that expresses peptide MHC to single-chain trimers. So this is a cell-based approach which relies on a co-culture between Jurkat T cells that are transduced with the TCR alpha beta that are obtained from patients. And in this co-culture, something striking happens. There is something called reverse thrombocytosis. So basically, these antigen-presenting cells capture the TCR in a highly specific manner. And these TCR positive K562 cells, and we have already did the testing in the system that we know the specificity for. So basically, this K562 T cells that capture the TCR can be sequenced to see what single chain trimer that they encode. And this gives us the information about the antigen specificity of the TCR. So this is work in progress. And we are really trying to understand the TCRs that are derived from pancreas tissue itself and draining lymph node itself, and hopefully we will be able to tell more about it in the coming years. And I would like to state again, understanding Treg biology and specificity are two key elements for successful therapies. Treg therapy, any Treg therapy, in my opinion, because we don't want off-target effects that can be quite detrimental on the autoimmunity side or cancer side. And with that, I would like to thank you, first of all, for keeping up with this talk for 1 minute more than an hour. And I would like to also thank my group, quite quickly expanding. I run my lab in close collaboration with my husband's lab. So this is my other half scientifically and at home. So we try to tackle all these questions as a joint effort. We are a happy, jolly group, and I have many collaborators I'm really thankful to, and I am happy to take any of your questions.
Thanks, Billur. That was really, really fantastic. So I think we still have time for a few questions. So there's a lot of comments just saying this was a really, really beautiful talk. So one of the first questions up there is about whether you see Treg stealing peptide MHC that have endogenous self-peptides loaded, or if it's somehow limited to foreign antigen derived peptides.
That is a very good question. Unfortunately, our tools are very limited. So what we have done so far is adding peptides from outside. So if you're answering the question of whole protein, we haven't done that experiment, but I think it is doable. So we can give whole protein and then see whether we pick up any of the restricted residues that we can detect on the dendritic cell surface that also is transferred to the Treg surface. But we haven't looked at it yet. I think it's an important question, if I understood correctly.
Yeah, and then so there's another question here. Said your images are really, really beautiful, and it looks like the activated T cells have more microvilli and bigger sizes than the naive T cells. If you had, just wondering if you had any comments on this, or if you know why that is.
So I wasn't super surprised about it because this is the blasting, blasting that we see in flow cytometry. So whenever you activate T cells, they go increase in forward and side scatter, and which we call blasting. But it happens quite dramatically within a couple of hours, which is kind of intriguing, but I wasn't super surprised. So yeah, they rapidly increase their protein, pumping, and then increase in size and active cytoskeleton microvilli. I think it's quite important in forming the microclusters. So if you give them enough time, they would really put their tentacles on dendritic cells and start, doing other things.
So I'm going to give you combine two questions about LAG3 together. So the first part is whether you think LAG3 allows class 2 stripping in a non-antigen specific manner. And then kind of related to that is whether TRex from 129 mice exhibit similar functionality, because the LAG3 alleles are likely co-inherited with the 129 CD4 allele, which has a number of SNPs compared to B6 and could affect class 2 binding. So I guess wondering about how this all works in different backgrounds that have different alleles of these genes.
That's a very good question. So as I said, we are limited by our tools in looking into the specific peptide MHC2 uptake. But that's right. So in different backgrounds, so far, I have looked into especially K haplotype backgrounds, because I had the antibodies, the HEL and mod cytochrome C. And unfortunately, IAB, like black 6 background haplotype, IAB haplotype of MHC2 has a problem in this reagent. So you can never get the peptiding group antibodies. But there is one which is EA specific. So there is an endogenous EA that is derived from MHC2 itself. So okay, it's a self peptide which is loaded to IAB. I did it with that too. And in that system, it worked exactly the same. So again, the TRegs captured, but I couldn't do it both ways because there was only one in IAB background. So in single way, it worked the same way.
Sure. And then what do you think that the LAG3 will allow antigen, like non-antigen specific stripping?
In my hands, because I was able to do, okay, so I didn't do that experiment, but in my opinion, so if I create the 2 antigen system again, LAG3 knockout and otherwise, and test it, I don't know the answer to that. My gut says it will still be antigen specific. It is not what is giving the antigen specificity. It is rather giving what the ability to pull. But I don't know the answer to that. I am hoping to test it. It's a very good question.
And then we'll just take one last question in the interest of time. Just wondering if there's a correlation between the affinity, the TCR affinity, and Treg functionality. But obviously, I think you showed nicely, right, that you can match the effector and Treg TCR and it works. But if you tried like a whole range of different affinities or dwell times even, right, that's the thing I think that's been shown to be really different for Tregs, whether that dwell time matters for this peptide stealing, peptide MHC class 2 stealing.
That's a very, very good question. I have some preliminary data addressing it. I haven't presented it here, but it seems to be with our limited understanding, because tools are really limiting us. So you can use altered peptide ligands, form the system that way. I am now doing it with OVA because OVA residues, the TCR binding residues, are nicely, laid out in the past, and you can just, create OVA variants and ask the question that way. It seems to be dependent on the affinity. So as the affinity increases, Tregs are pulling more. But this also brings the question that Tregs, well, the inherent affinity, TCR affinities of Tregs, probably, in the periphery, talking about the autoantigens, they have higher affinity for self-antigen than the clonal escape. Well, clonal escapees would be the highest, but, they are deleted. So Tregs may have the upper hand. It's not really an affinity situation, though. But, if you get an autoreactive... It's
more about having higher affinity than like true antigens.
Yes, like if you look at the repertoire, in general, they are like, the dogma, like mid affinity. But because we are able to make designer Tregs, and I feel that with my limited understanding, if we make them high affinity Tregs, it would be better in terms of making them more useful in capturing.
Okay. Well, with that, I just want to thank you again for a really outstanding seminar and looking forward to seeing where all this work goes and looking forward to everybody joining us in a couple of weeks for our next seminar. Thanks a lot, Billur.
Thank you very much.